Okay, this is James Leo. I'm doing lesson 13 group problems, questions 1 and 2. Question 1A asks us to indicate the banding pattern for the 4 DD-NTPs. And this is what I got right here. For DD-GTP, we have, so including TAGT, we have 10, and then we have 13. And then for DD-ATP, we have, including TAGT, got 7, 8, and 12. And then TTP, we got 9. CTP, we got 5, 6, and 11. These are indicated right here, as you can see. 10, 13, 8, 7, 8, 12, 9, 5, 6, and 11. For question 1B asks, what will happen if we use DNA polymerase 1 instead of TAQ polymerase? Well, what will happen is the DNA polymerase, because it's not TAQ, it will be denatured, and so it won't function because of that. The question C asks, what will happen if we add DNA helicase to what will happen if we add DNA helicase to the reaction? Well, it will still work. It won't really be affected. 1D asks if we add DD-ATP to one of the 4 tubes, or if we get to add DD-GGP, DD-TTP, DD-CTP to the other tubes, what's going to happen? Well, the reaction will fail because the sequence requires all 4 DD-NTPs. Question 2A asks, during typical PCR cycle, the third step, I have all the answers indicated right here. The third step is done at 72 degrees Celsius, while the second step is done at 55 degrees Celsius. B, AMPR is a common selectable marker that is found in many plasmid vectors, while restriction enzyme recognize and cleave DNA molecules at palindromic sites. C, during Arago's gel electrophoresis, DNA is placed in an electrical field. DNA migrates toward the positive electrode during electrophoresis. D, DD-ATP lacks the 2' OH group and the 3' OH group. E, transformation is a term used to describe the process where bacteria take up DNA molecules from the environment. F, the answers right here will be digested chromosome DNA is commonly used to induce the expression of the lac operon, while excal is cleaved. So here are the answers for questions 1 and 2.
Hey, it's Dylan. I had problems 3 and 4 in week 13. And so problem 3 says you are interested in using PCR to amplify the region of a double-stranded DNA fragment from E. coli. The sequence to be copied is located between the sequences in bold. And so we need to write the single-stranded DNA sequences that we would use for the primers. And so my primers I used right there, GCC, TTCA, and I labeled 5' to 3'. And the main point is to make sure that you are able to, the primers should encompass this DNA. So the first primer should be here and the second primer should be on that side. And then they use that mathematical equation to find about the temperature. So my 2 temperatures with my math right there are going to be 64.9 and 57.3 degrees. Each PCR cycle contains a DNA synthesis map. What are the sequences of the 2 new synthesized strands if DD-CTP is accidentally included in the PCR experiment? So basically what would happen is you'd have the sequences GA, TT, AGC, and then C would would stop the sequence. And then the same thing with from the other side of the primer, you'd have the TTATC 5' to 3', that's stopping the synthesis. So then when we learned about DNA replication in E. coli, we learned about DNA-A and DAM. Why are these two proteins not included in the PCR? So basically, the DNA-A was used to unwind. It would promote unwinding, is how I phrased it. And then the DAM it was used to tell the cell when initiation should start. So basically, we use the heat to unwind it. It denatures the proteins, and so it unwinds. And so you don't need the initiation to be controlled by the cell, because we kind of make that ourselves. Now for problem 4, we're given these restriction enzymes and the fragments they're produced. And so the first step was just the agarose gel image. And so it's pretty simple. Wherever there's a DNA fragment, you would include it in that lane. And then it gave you the some were multiple restriction enzymes at once. Draw a linear restriction map. So again, you use based on the agarose gel where it was cut. You can see that where BAM cuts, and you look at these overlapping, the cuts that are in between, and that's going to give you the spacing. So it's going to be 500 base pairs, then BAM, then 100 base pairs to HIND, 100 base pairs to ECO, 150 base pairs to BAM, and then 50 base pairs to the end. So now part C, we can see where it was cleaved at. It says what are the predicted fragment lengths if this linear molecule is cleaved with all 3 molecules simultaneously. And so these are where the cuts occur. Now that is the end.
Hey guys, here we have the week 13 group problems, and we're going to do 5 and 6. So 5 and 6, you are interested in studying a gene called your favorite gene that you have identified in this yeast. Detail the steps that you would use to generate cDNA and clone it, and use blue-white screening to identify plasmids that contain your favorite gene. So to create it, you're going to isolate the mRNA, add the poly-DT primer, and add DNTPase, however you say that. And then you're going to add these to splice it all back together, how we already know. So the cloning process, you're going to cut the cDNA that you've just created and the plasmid where you want to insert it, and you're going to mix the two. So use the DNA ligase to create the cDNA and insert it into the cloning site. And simply white colonies on the agar plate will have the inserts, where blue colony will not have the inserts that you've cloned. And for number 6, we were told to kind of just do our own research on CRISPR-Cas9. And so I'll just read you the paragraph that I wrote. CRISPR-Cas9 is a new tool used by scientists to cut and edit a genome and create many copies of the edited genome of prokaryotes. There are other ways to do this, but this method has proven to be by far the fastest and cheapest way to create many copies of the same DNA sequence. It works with Cas9 being used as a splicing tool to cut the genome of certain bacteria, which we can then insert our own edited genes and create millions of copies. Many uses include creating genetically modified crops, as well as cancer treatment in humans. All right, thanks guys.